curriculum development, including NTA and QCI accreditation process. Dr. Majumdar possesses a strong academic background with a PhD in pharmacy, M.Pharm in pharmaceutics from the University of North Bengal, and B.Pharm from BPUT, Odisha. He is also fellow of the Institution of Tennis, India, and holds an executive MBA. He has an impressive research profile, having published about 54 plus research articles in reputed international journals such as Elsevier, Springer, and Westlands. He has contributed to several book chapters and holds 3 Indian patents, including one granted by the Government of India. As an active researcher, he has supervised more than 25 plus M.Pharm students and is currently guiding PhD scholars. Dr. Majumdar is the principal investigator of the ACRB ANRA funded academic initiative 2025. His research interests include novel drug delivery system, colon-targeted drug delivery, multi-particulate systems, and nanoformulations. He actively contributes to academia as an editorial board member, reviewer, PCI inspector, and QCI assessor. He has also organized and chaired multiple national and international conferences. Now, Professor Bachar, are you hearing? Are you hearing? Your sound is okay?
Yes, I'm here.
Okay. Then we are going to start your deliberation. I am handing over the mic to Professor Majumdar to read out the small CV.
Good afternoon, everyone. It's my privilege and honor to serve as the chair of the session at international conference which is held in School of Pharmacy, SEACOM SKILLS University. I extend my sincere thanks to organizing committee members, those are inviting me. So today, this session, today our renowned speaker, Professor C. Bachar. So he is currently serving as a professor in the Department of Pharmacy at the University of Dhaka, Bangladesh, and has several prestigious leadership positions, including Pro-Vice Chancellor Academics, Dean of Faculty of Pharmacy, and Head of the Department. Professor Bachar received B.Pharm and M.Pharm degree from University of Dhaka, as well as he received his PhD degree from Jadavpur University, Kolkata, West Bengal. He has over 39 years of extensive experience in teaching, research, and academia. He is the highest respectively medicinal chemist with significant contribution in the synthesis of bioactive compounds, natural products research, and pharmacological evolution of traditional medicines. He has played a pioneer role in establishing clinical and hospital pharmacy practice in Bangladesh and has been actively involved in pharmacovigilance and antimicrobial resistance mitigation. With more than 140 research publications in reputed journals, his contribution to pharmaceutical science are truly remarkable. I warmly welcome Dr. Bachar to share his valuable insight with the audience. Sir, please start your session. Thank you, sir.
Thank you very much, Dr. Majumdar, and the panelists, and there's faculties and students of this international conference organized by SEACOM SKILLS University, Bolpur, and this is my very happy to be here today in presence of all of you. And this screen is visible. The screen is
Yes, sir.
Screen very good. Okay, thank you very much. So, very good morning to all of you. Good afternoon from Dhaka and good morning to from Liverpool as well as from all of the world who are playing in this conference. So, according to the request of the Professor, Associate Professor Dr. Praveen Banerjee, he requested me to keep in touch with the theme of this international conference of the emerging technology in drug discovery, delivery and diagnostics. And because of some affiliations, most of the cases of this my natural products, I designed it to make it in emerging technology for plant-based drug discovery. And you know that this integration of computational biology, molecular docking, digital technology is the next generation phytopharmaceuticals, because this terms is very much announced and resonance in the research community nowadays. So, as his request, I set this terminology as a title of this
Recording
research
in progress.
showing, and the theme of this lecture is on artificial intelligence, molecular docking, phytochemical informatics, system biology, digital phytomedicines, and precision natural therapeutics. And in my talk I will try my best to keep in touch with these relevant topics in this session. Before going to the details of this natural-based emerging technologies, and as speaking to my experience led by Dr. Rana Majumdar as an experience as a traditional medicinal chemist, actually what we do before, it is late 80s or before, or late 90s, early 90s, it is also in the market selling that. As the same is sorry. So same the change of this double bond to the single bond to make it in non-derivatives. So 6-chloro-5 cyclopentanyl is cleaned up. The non-carboxylic acid synthesis is also in the market. So on the basis of this, it is my lab is designed to synthesize a good number of carboxylic acids. I am confirmed. So that is the my laboratory in the Dhaka University, and as well as in the Dhaka lab, we designed a good number of compounds regarding this matter, and just going through because it's not for target of these things. So this is the scenario of the things which are actually doing things, and all of these activities we found some positive intellectual activities of the compounds, and it was reported in reputed journals of this Review Journal of Chemistry, Medicinal Chemistry, also the tetrahedrons. All of my students and my research students reported these things. So actually, about all these things, we are doing research on the basis of the receptor molecules targeting the respective disease pattern, and we had to synthesize a lot of molecules. But now, in terms of this emerging technology regarding the AI tools, these times and cost is the very much questionable in terms of reductions and as well as the evidence of these findings of the research things. So traditionally, drug discovery, most of the cases, we are following the disease-related genomics through target identifications, target validations, drug discovery, lead optimizations, preclinical studies, in the animal model, clinical in the human model, and then final approval for marketing authorization in the market. From drug identifications to the preclinical studies, that can be a good approach to using this computer-aided drug discovery, that is we are calling AI, artificial intelligence, making this formation in the bioinformatics, homologous modeling, reverse docking, target drug ability tools, compound design, library screening, docking, pharmacophore, and user molecular docking, dynamics, QM by ML, and finally this ADMET predictions or off-target predictions. That is the main use of this AI-driven drug discovery. And applications of this AI drug discovery, mostly as of the portions I showed in this target identification validations, in this traditional system, most of the cases we are following these things, but when we are using this AI genetics within the computer systems, it is very much specific to identifications and validations of the target. And after that one, we have to go for screenings of the lead discovery of the drug through this approach to make it a user activities. And then optimization and designs through AI tools for these target molecules. And this whole the target molecules almost give to optimize these things, that we should have to conduct a clinical studies in most of the cases, but all of these things is a dry lab, not in using any wet lab. So this AI-assisted drug discovery for hyperconstituents is through predicting drug-target interactions. And this artificial intelligence leverages machines and deep learning to predict drug-target interactions and facilitate the discovery and prioritizations of plant-derived phytochemicals as potential drugs. So here, these two slides almost saying the same things through pharmacophore model, then combinatorial, this compound library isolations, and this molecular docking, then homologous modeling sections, raw data, and this finally the predictions of these things. And this is usually now worldwide. It's a matter of things to discuss about this matter for lead optimizations and finally development of new natural products for optimizations. But why this plant-based drug discovery is re-emerging? Because natural products remains a major source of modern drugs. And most of the case, it is mostly these data-driven facts. We all know that a good number of approved drugs in the market originate directly or indirectly from natural products. So many examples are there: detoxins, digitoxins, aspirin, so many things are there. Anti-cancer drugs. So plant metabolites provide enormous structural diversity, impossible to replicate synthesis. Because we saw in the traditional way of synthesizing the drugs, we have to synthesize each and every pharmacophore or active constellation predicting all these things. But in terms of natural products, it is very much impossible to synthesize all these things because it's a vast number of structural diversities is there. So even then, this plant-based drug discovery may be the emerging things regarding this arising of antimicrobial resistance. And we all know about this coronavirus section things where the predominant activities we have observed by natural remedy through natural natural resources applied in the respective formulations. So current technology now is shifting from the traditional way of evaluations of the traditional medicines. So that's why it's being replaced by AI-guided compound prioritization, ultra-large virtual screen, network pharmacology, and multi-omics integrations. So scientific insights in this product space is modern drug discovery is shifting from trial and error hardware testing towards predictive computational pharmacotherapy or pharmacology. So here the traditional or historical evaluations, most of the cases is traditional medicines to AI-assisted pharmacomedicines through bioassay and molecular docking. Here, some examples of working compounds through which we have to define what drug is specifically for the target molecules on the basis of the data collections and curations. And then the compound is targeted from according to the design and criteria. And after that, using these AI techniques through molecular modeling, area predictions, bioactivity predictions, AI predictions, and drug target structure, we have to find the drug target using AI model selections. And when the drug candidate is selected, and the pharmacophore model docking, we have to go for human relevant assay. This part we have to think about this dry lab, and then we have to go for chemistry design and synthesis for the wet lab. Then after that, go for preclinical and clinical trials for approval to the marketplace. This is the usual practice for AI drug discovery. But AI in phytochemical discovery is very much interesting nowadays. Artificial intelligence for bioactive compound predictions is a current challenge. Technology is used most of the things. It's machine learning, as I told you before, deep learning, graph neural networks, transformer-based molecular predictions, and the applications of this AI is to predictions of drug-target interactions, identifications of hidden pharmaceuticals, phytochemicals, ADMET predictions, and drug lead optimizations, and finally automated literacy mining. And the research impact is this AI can screen millions of phytochemical detections in hours compared with years of laboratory, which is required because I have shown you in the synthesis approach, years of inputs we have given a number of only 30-40 compounds. But that can happen in few hours with this AI-driven techniques. Molecular docking and viral screening. Computational predictions of plant compounds pertaining interactions is very much important discussions today here in respect of natural product discovery, which is followed by binding affinity predictions using molecular docking analysis, drug target interaction analysis, binding site characterizations, structural bed design, and following advanced computational tools, which is followed by Autodoc, Stranger, DeepChem, AlphaFold assisted docking, and AI native docking frameworks. And modern advance of these using tools are these ultra large scale docking can screen billions of molecules of computationally before wet lab testing. And this research significance of the molecular docking and virtual screening is its cost is very much reduced, as I told you before, and experimental burden is reduced, as well as drug development time is significantly reduced. Here, there are some examples of the molecular docking reduce the potentials of clean amylocarpa, where one of the most promising compounds is found is chemotherapy 6. This one is docked into the active site of COVID-19 protease. This structure, this camphor is docked into these active sites of the protein COVID-19 protease in terms of these amino acids, and these two figures, this mapping surface showing camptothecin 6 occupying these active pockets of the protein molecules, so these receptors, and the 3D positions of the camptothecin is this one. So this is the way we can use the molecular docking for predictability of the active molecules for a specific therapeutic purpose. So online database and digital informatics is very much emerging tools for accelerating natural drug product research. Most major databases are PubChem, Zinc, ChemEMBL, TCMID, KEGG, and PlantNet. These are the source of the major database from where you can get natural products as the library. And emerging technologies, these are usually known as cloud-based collaborative research, real-time compounding, sharing, AI-assisted mechanical toxicological mining, and digital herbal fingerprintings. And you know, this nowadays collaborative research is very much important to enhance this number of molecules, like India is doing this traditional knowledge data library, because this traditional knowledge regarding these molecules is very much important to enrich these things. For this we can use for future perspectives of identifying the molecules. The online database and digital informatics importance is this online scientific communications enables global collaborations, faster validations, shared computational resources, and online access to molecular libraries. And key message of these things is plant-based drug discovery is becoming increasingly data-centric and globally networked. Now, the multi-omics and system biology. And beyond the single compound, systems level understanding is being predicted by integrated technologies, by genomics, transcriptomics, proteomics, metabolomics, and network pharmacology. As well as using these omics with the tools of this AI can identify synergic compounds, predict pathway of modulations, discover hidden therapeutic mechanisms. So this is the very important tools on the basis of the multi-omic systems using AI tools. Why it is matter? Because medicinal plants contains complex mixtures and that act on multiple biological pathways simultaneously. So that these tools can make you easy to identify the compounds with possibilities of respective pharmacological activities. And future predictions of these things is digital twin biological model may predict personalized response to plant-related therapeutics. Very important things is precision in regarding this personalized response of these things. And these are examples of this how these precisions for the identifications of these data-driven interpretations of the respective molecules using personalized medicines. Because that's why we have to collect samples, living samples from the persons, the sample preparations of details, and then data independent acquisitions and LCMS and MS. Then we have to optimize these things for data-based analysis using UPLC QTOP MS, and then have to identify for identifications and the data interpretations. So this is how we can predict the precisions of these AI-based technologies. So what is the futuristic approach of this plant-based drug discovery? And we are expecting that this hypothetical future workflow will emerge after 2035. So, an ethnobotanical surveillance will enhance the satellite-guided biodiscovery mapping, leading to anonymous robotic extractions, then to quantum AI module simulations, data, digital twin efficacy predictions, and finally personalized phytopharmaceutical manufacturing. And we are expecting that today a number of good young scientists and the young college, those who are involved in such types of national product research, we are expecting that we are getting good kinds of contributions from India and all over the world regarding this matter. So the future technologies using these things is quantum computing in docking. These will be things, how we all know that India has gone and steps on quantum computing for 25 capacities, and within 2 or 3 years we are expecting that India is going to steps in 100 capacities of this quantum computing, so establishing a great achievements of the development of the India in research and this development of the total scenario and GDP of these economic perspectives. AI generating phytochemicals analogs, self-learning molecular design systems, blockchain authenticated herbal traceability, and personalized plant-derived therapeutics. And the visions of these futuristics is the future phytomedicines may evolve into precision pharmaceutical engineering. So, when there is a futuristic approach of this AI-generated drug discovery, but there is some challenges in the field, because on the basis of the scientific and technological variance. What are the major challenges are variability of phytochemical composition, as you mentioned, because on the basis of these things, because on plant materials, on the basis of the locations and geographical locations, the number of plant metabolites is very much variable. So it is very much difficult to accumulate basically these plants for the applications of respective diseases is actively working or not. That is the things which have to be challenged, have to satisfy this challenge. Lack of standardization extractions. That's why we are not getting the same number of molecules. The same plants in India, a good number of other metabolites is coming. When we are doing in Bangladesh, with another types of, because of soil, environment, we know all these things, but there are some variabilities there. As well as that's why the reproducibility is a matter. The poor reproducibility is there, and limited curated database, that's data quality and availability, and AI module bias, difficulty in biological validations, and regulatory matter is very important to get approval for before going to the market, because if it is not able to solve these regulatory matters regarding the violations of these things, it will be a great challenge if it is very much effective to the human body. The discovered or lead compounds as well. So complex multiple component interactions is very much in this case of this, because one compound is multiple components is interactions. This is very much difficult to predict which compound is effective with which disease, because according to our previous studies, we have seen that the single molecules is actively so many types of diseases, and that's why interpretability, interpretations, and transparency of such kinds of discovery of the natural products may be very challenged in near future. At the same time, critical concern of these things of computational predictions is in vitro validations, in vivo testings, and clinical trials. So we have to meet some strategy needs. For this progress requires the global data standardization, very much important to solve these problems, and integrating omics database, AI governance frameworks, and strong academia industry collaborations. Through these things, I think we can solve these problems of this challenge in these fields. Finally, the future drug discovery is computational, predictive, and plan inspired. So through these things, the key message of this today's discussion is AI is transforming pharmaceutical drug discovery, molecular docking accelerates target identifications, multiomics enables systems level phytopharmacology, and digital collaborations in globalization's medicinal plant research and future medicine, phytomedicines will likely become highly personalized and precision-driven. Finally, emerging technologies are converting traditional medicinal knowledge into data-driven precision strategies for the future of global healthcare. Thank you to all of you, and Dr. Nazneen Sultan and Dr. Kakunan is my contributions in designing this lecture today to all of you. Thank you very much for giving me the opportunity, and thank you to the Ravi Mukherjee, Dr. Deepak Barunja, and all of you, those who are present today in this conference. Thank you very much, and regards from my personal regards and from the Dhaka University as well. Thank you very much to all of you.
Thank you, sir, for your valuable speech. Sir, you enlightening the session and valuable insights into the various AI-generated molecular docking as well as the applications of AI in drug discovery. It's have truly enriched our understanding and inspire us to strive for academia as well as the industry. Hope all are enjoying this session. Thank you all. I conclude. If any questions from the audience, please ask me. Anyone from audience?
Hello.
Yes.
Myself, Dr. Manoj Sharma. I have done my PhD in cancer biology. So I would like to know, you told about this AI and which software and how will apply this AI in pharmacology?
I have mentioned some things you see.
Software, yeah, I have seen, but where do we get this software?
Oh, that is the things, because most of this database are have to collect from the this valid sources, because most of the things pirated database is there, but most of the cases we have to collect from the respective program design, those who are contributed in these things, because it is very much costly. This most of this database are. That is the thing, because that's why we have to, that's why I told you collaborative research is very much important. So Jadavpur University has under the leadership of Professor Mukherjee, that have a data library. They are contributing with other international bodies for optimizations of certain tools. So that's why we have to collaborate with the research of using such kinds of tools to enrich the database. And each and every institutions, those who are analyzing such kinds of library, compound library, and have to share these things. So with this, using the software, and that is very important. And software must be available on the basis of the software companies, those who are on the basis of need of this researcher, because they are designing this research data library on the basis of the need of the researcher. We are asking to them to know how to do these things. So we have to, because molecular docking, this ADME predictability, all are have got some softwares. So these are the tools for AI-driven research. That is the thing. But we have to find it, because a good number of AI tools are available regarding this matter.
Sir, one last question. If I want to collaborate with you, what is the procedure?
I have a good scientist, as I mentioned in the last slide, Dr. Nazneen and Dr. Nag.
Yeah.
They are the medicinal chemist and the biological, they are all renowned scientists. They are the persons who developed these COVID vaccines in Bangladesh. You know about the Bongo vaccine. So they have, because they have some accessibility of such kinds of data, and they have some also some softwares using in these regards. So, because to all of you know all of you that I am going to retire with a very short period of time from the university, but also actively involved in the research. So communicate with us regarding your topics and things that we can, so we can extend your cooperation to you.
Thank you.
Definitely.
Thank you.
Thank you very much. Thank you.
Thank you, sir, for your valuable interaction.
Mhm.
Anyone from
Thank you very much.
audience? Any questions? Any questions? Okay, sir. Thank you for your answers.
একটা আছে
Okay,
question.
Thank you, Dr. Ranavir. Thank you very much.
Sir, one question is there.
Yes, please.
Hello.
Yes, yes, sir.
Okay. My
Yes, sir.
question is that at present, the pharmaceutical manufacturing companies facing
Yes.
the major problem, that is the toxicity and drug adverse reactions.
Yes.
How this AI will help and how much it will be correct? Have you any idea?
Because these traditional manufacturers mostly using the whole extract, and these whole extracts for detoxifications they are using some special heavy metals and other things, and the traditional way they are approaching to the formulations that is not purified exactly, and most of the things, the side effects of this impurities presents for the process of the optimizations of development of the products. And these interactions mostly not for the phytopharmaceuticals but originate from the other things, excipients and the process of the evaluations of the traditional medicines, that is Ayurveda and Unani and other things. So that can be reduced when we use these AI driving tools, optimizing the specific methods of extractions. As I told you before in the lecture, that is a challenge in the for these purified or the specific method of extractions, and that have to be optimized on the basis of the program number of molecules which are actively effective to the respective disease pattern. And if it is possible to identify the specific tools for the extractions of the active biomolecules specifically for the respective disease, then the side effects of the preparations will be reduced definitely, I believe. My experience says like that.
Thank you.
Thank you very much.
Just one question.
Yes, please.
Man and machine, there is difference.
Yes, yes,
There
yes.
should be something. As a past
Yes.
regulator, my question is, how the regulatory authorities will solve this problem? Who will be responsible, man or machine? This is my question.
I mentioned in my challenging of these AI tools used in the drug development, because these AI regulations, the regulatory approval is very much important, because we have to convince the regulatory matters regarding the evidence of this establishment of the new drugs, because the respective tools that should be validated regarding the toxicity, predictability, and target optimizations of the molecules through the respective method of this AI driving tools for establishing the drugs. And if it is able to predict these things through preclinical and clinical trials, and that can be an evidence to the regulatory for the safe use of the medicines for the community. That can be done because proved this evidence in terms of preclinical and clinical studies and the data, because all the tools used in this AI driving is data driven. Data is there. So that data will help the regulatory bodies to decide the drug is safe or not. So I think it will be very much difficult because machine, and that can be validated with in vivo and in vitro both of them. That can be evidence, and this data should be submitted to the regulatory body for the convenience, for getting approval for marketing authorization. I think it can be possible. May I please, sir.
Thank you.
Thank you very much, sir.
Our Principal Professor Ravi Chandra wants to ask you a
Yes.
question.
Yes, sir. Yes, sir. Yes, sir.
Good afternoon, sir.
Good afternoon,
My
sir.
one query is there, that is your suppose drug interaction. It is a problem.
Yes.
That is that a person, suppose simultaneously he or she is taking Ayurvedic and modern medicine. At that time, drug interaction is a major problem.
Yes, it is evident.
And can AI help to identify drug interaction?
Drugs, herbs interactions, that can be, I think, these tools, because when the drug interactions, drug and herbs interactions is another thing when we are taking the body, because these interactions is mostly based on the receptor side. And this receptor is predicted by AI tools. When we see that these conformational structures of the modern medicines and this traditional medicine is similar, then there is a competitiveness of the liking molecules to the receptor sites for exhibiting its pharmacological actions. So, on the basis of these interactions, due to drug and herbs mostly the target these active molecules because of the receptor bindings. So that is competitions that may lead to increase the half-life of the drug. So when the half-life is drugs, that may, in terms of narrow therapeutic drugs, that may lead to toxicities, because narrow therapeutic drug you know very well, so it will go to toxicities. So that may then happen, and that predictions of the receptors can be identified with the use of AI tools. And I hope that can be solved, these types of interactions with modern medicines and herbal in combinations on the basis of the persons, those who experience these side effects can be analyzed with these blood samples or these receptor site from the target sites about these things can be predicted, this interaction, I think. That the future will say, our young students, they will work in the near future, I think. I think my prediction is that it's possible to identify.
Thank you, sir.
Thank you, sir. Thank you, sir. So many things.
Before closing the session, Professor Prasad,
Yes, sir.
Bangladesh will
So when I will be in West Bengal or Kolkata, then I will definitely visit SEACOM University.
Yes, that's why.
It is very nice to see all of you there.
Okay. So যখনই কলকাতায় আসবে একবার
অবশ্যই
তারপর
অবশ্যই আমি সেখানে হাজির হব। One day I will give a interactive sessions with the students and the colleagues in the SEACOM University. I will be there.
তোমাকে তো আমরা কিছু দিতে পারলাম না, তুমি নিতেও চাও কিছু, কিন্তু যখনই
না।
কলকাতায় আসবে
আমি আসবো। আমি আসবো।
একটা পেমেন্ট করে দিতে চাই আমাদের কলেজে।
না, আমি আসবো আপনাদের সঙ্গে। আমি another sessions will be there.
আসলে আরো ভালো লাগে। ঠিক আছে।
আমি আসবো।
Thank you, sir.
Thank you very much, all of you. And thank you to our university, SEACOM University, for giving me the opportunity to share my views and ideas regarding this matter, the topics of the day. Thank you for all.
Sir, one certificate can be displayed. Sir, please.
Yes, sir.
Please be token of
Thank you, thank you.
appreciation given by the
Thank
SEACOM
you very
SKILLS
much.
University, sir.
Thank you very much, sir. I shall collect it physically.
Obviously,
from
sir,
professors
obviously.
and from directors. Thank you very much, sir.
ঠিক আছে
ঠিক আছে, বললাম।
আমাদের নিমন্ত্রণ রইল আমাদের ইউনিভার্সিটি আসার জন্য যে কোনো সময়।
তরুণদাকে দেখছি না তো।
তরুণরা অসুস্থ, তরুণদের কোনো আসবে
ও বয়স হয়ে যাবে স্বাভাবিক।
না, বয়স হলো আমার থেকেও কম, ও হচ্ছে 67, আমি 68।
ও ঠিক আছে।
তো আজকের মতো শেষ করছি।
হ্যাঁ, ঠিক আছে।
প্রফেসর রণবীর চন্দ্র, মাননীয় প্রিন্সিপাল স্যারকে অনুরোধ করবো
Ranavir Chandra is there, I think he was there. Dr. Nana Mujumdar can complete this thing.
Thank you so much, sir. Thank you so much to our session chairperson also, Professor Rana Mujumdar sir.
Thanks a lot.
I request our respected Associate Professor Prabir Kumar Banerjee sir to felicitate our respected session chairperson, Professor Dr. Rana Mujumdar sir. We are very grateful to Dr. Shritesh Bhaskar sir for his positive and enlightening session to be present with us virtually, and we are very thankful to Professor Dr. Rana Mujumdar sir to be present and making our event delightful. Thank you so much, sir. Thank you, Professor. We are ending our plenary session here, and very soon we are going to start our cultural session and lastly the valedictory session at second half. Thank you so much, sir, for being present with us.
Thank you very much. So I am quitting from the.
Okay, okay.
Thank you very much. Thank you.
Thank you, sir. I request everyone যে স্টুডেন্টসদের ফার্স্ট সেশনে পোস্টার প্রেজেন্টেশন আছে তারা কাইন্ডলি যেখানে তাদের পোস্টার প্রেজেন্টেশন ভেন্যু আছে সেখানে চলে যাবে।